This video is going to get you through the classification notes. One of the things that I want to stress as we go through this chapter, and I think I make this note at some point as we kind of go through here, is when Woese provided his information that helped to establish the 3 domains of life, it threw taxonomy into this whole topsy-turvy world where nobody can agree on which taxa means what, and it kind of reclassified everything, especially everything that was prokaryotic. And so what used to be a kingdom became a domain. There's a bunch of names that have had to change, like there used to be a kingdom Monera, and that kingdom no longer exists, and a lot of this stuff hasn't been ironed out completely yet. And so I'm not going to generally make you memorize a bunch of taxonomy since it's not ironclad and it shifts all the time, especially since the 90s it has been shifting a whole heck of a lot. So just kind of keep that in mind as we go through there. All of our taxonomy is hypothetical. It changes based on new evidence, and right now it's still just not very set in stone just yet. So, mentioned this dude's name just a second ago. Carl Woese used rRNA sequences to compare different organisms to each other, and what he found was when he created sort of a tree of life or a family tree of life, there were 3 branches on that tree. There was the branch that included all of the eukaryotic organisms, there was a branch that included the bacteria, and then there was a branch that included the archaea. Both of these guys are prokaryotic, but they are very different from each other, and so they shouldn't have been in the same kingdom because their rRNA showed they were not related to each other. And so he broke apart that old kingdom Monera and then added the taxon that was domain, and again blew up the taxonomic world. But it's good evidence that he used, and so we're still going to stick with that at this point. So to make sure you answer all the questions, what are the 3 domains? You can see those kind of in bold: eukarya, bacteria, archaea, plus we've talked about them in earlier chapters. Which 2 are prokaryotic, bacteria and archaea, and what evidence did he use? He used the nucleotide sequence of rRNA to help make this family tree. Next step, a bunch of terms for you guys to define, and the definitions are really easy to find in the book, but here you go. Taxonomy is the science that studies organisms and then arranges them into groups. Like if you're practicing taxonomy and you discovered a new species, you get to figure out, well, which family is it supposed to go to. If it's something that's dog-like, you would put it in the family Canidae. If it was something that was cat-like, you would put it in the family Felidae. So you just put things into groups based on who they're most related to. In the old days, they did that based on appearance. Nowadays, we're using DNA evidence and nucleotide sequences to get more specific in terms of who's related to who and then who should be grouped together with who. Identification is the process of characterizing an isolate to determine the group to which it belongs. So this is kind of what you're going to be doing with your unknown. You're going to get a broth that has some unknown organism in it, and you're going to have to identify that and then figure out, well, what kingdom does it belong to, what order does it belong to, and so on. Classification arranges organisms into similar groups, making them easier to identify and then study. So classification is what puts each organism into each taxon, essentially. This is the current taxonomic hierarchy. It starts with domain, and it has since the 90s. Underneath that you have kingdom. You would think the kingdoms would kind of be set, but they are not at this point. We are in the domain Eukarya. We're in the kingdom Animalia. Phylums. Kingdom is a group of related phyla. We are in the phylum Chordata. Class. A phylum is a group of related classes. We're in the class Mammalia. Class is a group of related orders. We are in the order, I have to think about this, Primates, because we have opposable thumbs and forward-facing eyes and so on. An order is a group of related families. We're in the family Hominidae, as are anything that are human-like. Families are a group of related genera. We're in the genus Homo. Like Homo sapiens, Homo neanderthalensis, there's a bunch of extinct homos that used to be running around, but right now it's where there's only one extant species that is still traipsing around that is in the genus Homo. And then genera is a genus, is a group of related species. We're in the genus and species Homo sapiens. And so we talked about this in earlier chapters, but make sure you know how classification works and why an organism goes into each particular group. Nomenclature is what you have next. Nomenclature refers to names, and so again, if you were to discover a species, you would have to assign a scientific name to that species. That would be based on which genus and species it got grouped into during classification and taxonomy. What are some things that can be done to identify microorganisms? And again, you're going to be doing this in lab, so in theory you should have some ideas already, but You should put it under the microscope after you've gram stained it and look at it to find out is it gram positive or negative. If you think it might be a mycobacterial species, you can do an acid-fast stain instead. But just looking at it under the microscope, you can start to weed out things it's not. For example, if you look under the microscope and it's a gram-negative rod, then you can cross off all your gram-positive cocci and all your gram-positive spirillas and all your acid-fast bacteria as well, for that matter. And so it helps you start to narrow down what species you're looking at. So microscopic examination and differential stains can help. Culture characteristics, that's when you start to look at colony morphology, like is the colony raised, entire, convex, rhizoid, lobate, all of those terms that you learned in lab. For example, if you were to see an organism that grows red circular colonies, then you would know it's not Micrococcus luteus because it doesn't grow red colonies, it grows yellow colonies instead. And so again, it helps you weed out what it might be and point to what it might be at the same time. Biochemical tests, that would be like your citrate, your MRVP, your phenol reds, all those tests that we've been running in lab that provide you more evidence that can help group the organism into a particular group. Now, nucleic acid analysis gets you a point-blank answer. If you run like a barcode analysis on it, it will tell you exactly what species you have. We don't do that in the lab. It's expensive still to this day to run nucleic acid analysis, although it's much cheaper than it used to be. But it would just tell you what species you have and who it's related to. You can also look at diagnostic symptoms, like if it's a patient that's coming in and they've got a cough, well then you know it's probably not Staph aureus, because Staph aureus tends to do a skin infection thing and not lead to coughing symptoms. And so you can use the symptoms to also help you figure out what organism it might be. Alright, so biochemical tests, I gave you some examples earlier. Well, this is microscopic examination, not a biochemical test. This is colony characteristics, which is not a biochemical test. But this is that phenol red test. You can see a negative test, acid and gas production. This one made some weird stuff off over there, but that's a biochemical test, so C is the right answer to that one. How could you use this to help you identify a microorganism? Well, this is a fecal test, fecal sample. It's kind of funny when you think about it, but sometimes people have to go home and literally poop in a cup and then bring the cup back so that you can view it. And you can actually make smears with a fecal sample so that you can view what might be under it. Because they might have diarrhea because they had a bacterial infection, but it also might have been a protist infection. And so making that smear can help you do the microscopic examination of it. You can also do any of the other tests that go along with that I mentioned earlier. You can try plating it and looking at colony characteristics. You can run biochemical tests. You can do all that other jazz too. All right. Phylogeny helps to essentially put an organism onto that tree that Woes had started to make earlier. What it's looking at is the evolutionary relationship between one species and another. So as you're looking at this right here, ciliates can be more closely related to plants than they are to the slime molds because their branch point is closer to plants than it is to the slime molds way off over there. So this is showing you who's more related to who. Strains is a term that really only gets used in the microbiology world. They're called different things in the other like botanical and zoological worlds, but a strain is essentially a subspecies. It's a type of a species. Now, one of the strains that is kind of relevant when it comes to microbiology relates to E. coli, and so I have a little video that can help explain this just a little bit.
E. coli 0157. The CDC actively tracks outbreaks of this pathogen, which hospitalizes approximately 2,000 Americans every year. Karen Neal is an epidemic intelligence officer who works in the Outbreak Response and Prevention branch of the CDC.
Escherichia coli, better known as E. coli, are a group of bacteria. And most strains are actually harmless and live in the intestines and can be beneficial to health. Some strains, though, particularly E. coli 0157, can cause illness.
Because this disease initially presents like any other stomachache, an E. coli 0157 victim could already be in a life or death situation.
I'll stop it here. They just kind of go into it in a little bit more detail. But E. coli 0157, especially 0157H7, produces that Shiga toxin that we had talked about before that basically breaks down your blood vessels so you start to bleed a lot. Well, that's just one strain of E. coli. And like she said, there's a bunch of strains of E. coli, and most of them are harmless. But this one particular strain can be very deadly because it has that Shiga toxin that it can use inside the cells. I don't want to close that. There we go. After that, definitions. So a species is a group of strains, or it's one particular type of organism. This is E. coli. Again, E. coli is a species taxonomic unit. Everything from here on out on this list is a group of the things that came above it. So a genus is a group of species, a family is a group of genera, an order is a group of families, and so on all the way down. The other little subnotes that I want you to make as we go through here, find where family is on the list. It's a group of related genera. I said that earlier. In the microbacteria world, families are going to end in the suffix ACEAE every time. Order, that is just below that on your list, but above that on this picture. Excuse me. It's a group of families. It's going to end in the suffix ALES. ALES is how you usually say that. So if you see those suffixes, then you know which taxonomic group it happens to be talking about. All right. So this is our complete taxonomic hierarchy with a picture that I just like to look at because it makes me happy. This was my son back when he was adorable and cute, way back in the day when I would, give him the nice bowl haircut. He doesn't let me touch his hair anymore, but whatever. This is, again, how your book spelled eukarya, but that should have been a K. We are in that domain because we have eukaryotic cells. We're in the kingdom Animalia because we have to ingest our food, and we're multicellular and we're motile, and there's a bunch of other characteristics that put us into that group. We're in the phylum Chordata because we have a dorsal hollow nerve cord and a postanal tail and pharyngeal slits and a notochord. We're in the class Mammalia because we have mammary glands and hair or fur to help us stay warm, and we're endothermic. We're in the order Primates because we've got opposable thumbs and forward-facing eyes. We're in the family Hominidae because we're humans, and then this is human off down here too. All right. There are some informal categories that can be very useful. These aren't things that are taxon level stuff, but they help explain something about the behavior of a particular organism. So some of the ones that I want you to know. Lactic acid bacteria. Those are bacteria that are capable of lactic acid fermentation. We talked about that in the metabolism chapter, but you tend to find those in things like yogurt. They're going to have lactic acid bacteria in them. Anoxygenic phototrophs. This would be your nice purple bacteria off over here. They do photosynthesis, but they don't release oxygen in the process. Endospore formers. This is more than likely like Bacillus anthracis that makes anthrax. It's a spore former. Clostridium is a spore former, but Clostridium and Bacillus aren't related to each other. So again, it's an informal category, but it tells you something about them. They can make spores. Sulfate reducers is another group. They can, as the name implies, reduce sulfate as an energy source. And then one that's very important that isn't mentioned in the book, it has different names. Usually they're called enterics. That's E-N-T-E-R-I-C-S. Enterics are organisms that live in the gut of other organisms. Like E. coli lives in your large intestine, so E. coli is an enteric. Enterics can also be called coliforms. That's C-O-L-I-F-O-R-M-S. Coliforms are enterics that are contaminating water, and it tells you that that water has been fecally contaminated. And so coliforms are something that you see whenever you get a water quality test from a city or a municipality. Okay. Most of the information from Chapter 10 is going to be covered in lab, but it wouldn't hurt you to read it again. Pay particular attention to tables 10.3, 10.4, 10.5. Remember that you don't have to memorize anything in these tables, but it can be a good reference, especially as you're making or writing your unknown report. So this is 10.3. Bergey's Manual of Systematic Bacteriology is like the Bible of systematics when it comes to microorganisms. I'd love to show you a sample, but it's like the most expensive book in the world aside from some of like the jeweled Celtic books from way back in the day. But it at least does show you who's in which phyla, gives you some specific organisms. And again, you don't have to memorize any of this stuff, but it's a good resource to use as you're trying to write that unknown report. This is just a continuation of that. So proteobacteria is a really big group. This has a bunch of different little subgroups in it, but they're in there. This is the methods used to identify prokaryotes. We kind of already talked about all these things. So it's a good little table to study as you're looking back over. What was the question? How did I phrase it? What are some things that can be done to identify microorganisms? Well, these are the things you can do. How can size and shape and gram stain help? Again, it helps you weed out organisms that you know it can't be based on what you see. So it can also determine if what you're looking at is prokaryotic, like these blue cells here, or these small pink cells here, or is it eukaryotic, like these big old neutrophils in this case that have a giant multi-lobed nucleus going on. It helps you rule out the other species. So, for example, based on this gram stain, this isn't E. coli because E. coli isn't a diplococcus, and what we're looking at right here is a diplococcus. Same for up here on this one. It's usually showing up as a diplococcus, or every now and then a little chain of them. How can culture characteristics help? So these are two organisms that we use in lab periodically. The only one that we use that makes a red pigment is Serratia marzacin. So just growing it on a plate and you've got red colonies, that tells you something. Pseudomonas produces a green pigment that it kind of deposits into the agar, and so if you grow a plate and it's got green pigmentation to it after you've grown it, it's probably Pseudomonas aeruginosa. And so pigments that get produced, the shape of the colony, all of those things can help you, again, point towards a specific species and then weed out the species that you know it must not be based on those colony characteristics. Let's see, beta hemolysis in here could mean that you have what? So this is the beta hemolysis. One of the organisms that's capable of completely destroying red blood cells is Streptococcus pyogenes, which causes strep throat. So C is the right answer to that. So that's using a colony characteristic in a blood agar plate to help weed out some of the other things. All right. Let's see, I already did the how can gram stain be used, but just in case you didn't get stuff written down, it's distinguish between positive or negative species, and then help use the shape, the arrangement of the organism to provide you with some more evidence. How can culture characteristics help? Colony morphology changes, kind of just established that stuff earlier. Using the shape and size and pigmentation and margins and elevation and all that stuff can help. From here, we're going to talk about some of the biochemical tests. We did perform everything that is below that list in lab, but we just want to make sure you understand it in lecture 2. So catalase test, if we haven't run it yet in lab, this is where you drop some hydrogen peroxide on the organism and then wait to observe bubbles. If there's bubbles present, it means the organism produces catalase so it can break down hydrogen peroxide. So bubbles if catalase present, that's catalase positive. If there's no catalase in the organism, we would classify that as catalase negative and you wouldn't see the bubbling. So catalase positive, catalase negative because this side didn't bubble. This organism is pretty clearly catalase positive. Next up, citrate test. We get a color change. The tube starts out looking green like this one off over here. But if the organism can use citrate as a carbon source, then it ends up turning the agar blue and it grows on the surface of the slant. And so this is what a positive test looks like. Citrate test can also just be positive if you get growth on the slant but no color change. So you've also got to look for, is there growth, yes or no. But this is a positive citrate test. Methyl red is part of the MRVP test. It tests for the presence of mixed acids. It's positive if it's red and it's negative if it's yellow. Oxidase test. We use the dry slides for this one. You smear a little bit of organism on those cards, and then we observe for within 30 seconds is there a color change that occurs, and if there's a color change, it changes to purple, then the organism is oxidase positive. If it doesn't change colors, it's oxidase negative. Sugar fermentation tests are the same thing as phenol red tests. This is what a negative test looks like right here. This one produced acid and gas, so the derm tube has that little gas bubble that's inside of it, so you would write AG for this one. And then this one actually produced some ketones, and so we've got some additional stuff that was made that actually made the pH kind of change in a different direction, is how I'll say that for right now. Vogus Proskauer is the VP part of MRVP. If it turns red, it's positive. If it doesn't, it's negative. Remember, this is the one where you do have to wait a full hour for the test results in order for you to see whether or not it really was a negative test. This one is detecting acetoin, which may be present if we ferment it, and Enterobacter is one that makes it. This is just a summary of all those tables. I know I went through them really fast, but just kind of get down what's it testing for, what does a positive and a negative test looks like, and you're going to be okay. I didn't go over every single one of these. I just went over the ones that we actually do in lab. One of the other ones that I wanted to mention, urease right here. One of the characteristic signs for a person who has a Helicobacter pylori infection is it is a urease positive organism, and so it can break down urea and produce ammonia, and so you can smell ammonia sometimes on the breath of a person who has that ulcer. They call that a breath test. It's kind of using this urease stuff in order for that detection. Okay, I did actually say in my notes that I added urease to your test results, so then I also just gave you that little story, but you have that as a definition. I just didn't add a slide to it, to the presentation, but make sure you know what urease does. I'll look here. I've got it on the slide after all. I just skipped some stuff apparently. Okay. How do dichotomous keys work? I've given you one of these within your unknown reports paperwork. Dichotomous key is essentially sort of like those choose your own adventure books from back in like elementary school or middle school. Like you get to the bottom of the page and you have a question, and depending on which way you answer that question, you either go to page 16 or you go to page 64. Well, it's the same way with a dichotomous key. So when you do your unknown, the first thing that you're supposed to do is gram stain. And if it's positive, you would go this direction, and there's a bunch of other questions off over here. But if it's negative, you take this branch. From there, you look at what shape was it. If it's caucus shaped, there'd be questions off over there. But if it was rod shaped, next thing you would do is come down and do the oxidase test. So what you do is you just look at what your test result was and then choose that path, and you keep going until eventually you know, well, this must be my unknown because the dichotomous key led me down the path that led to E. coli down here at the bottom. So it's a tool that can help you identify an unknown organism. The way your book defined it was it's a flow chart that you use test results to complete. After that, I give you classification of microbes has changed dramatically in the last 20 years because of gene sequencing. While we don't do any of the genetic techniques in our lab, they are important for current taxonomy. I used to test over file names, but then all of them changed, and then they changed, and then they changed, and so I stopped testing over those because the name keeps changing, and until it's held still for like 10 years, I'm not going to teach them anymore. I did give you a couple of model organisms, Crenarchaeota and Euryarchaeota, that I wanted you to get down. One of the things I want you to notice is sometimes the name tells you something, like it says Crenarchaeota. Notice Archaea is right there in it. That means that's probably in the domain Archaea. Same for the Euryarchaeota over here. Notice Archaea is right there as well. So for the Crenarchaeota, would cellular morphology alone help you put an organism into this kingdom? No, because it can vary. So notice off down here on these guys, yeah, there's some rod-shaped organisms, but there's also some organisms that don't have regular shapes, and so you can't use shape alone to get there. What is the gram reaction of this group? They're all gram-negative. Explain the taxonomy of this group. This is, again, going back to who knows. Some people consider Crenarchaeota to be a kingdom. Some consider it to be a phylum. We don't really know. We have not settled on what characteristic or what taxon, that's going to be yet at this point. What we do know is this is one phylogenetic grouping because rRNA separates them from the other archaeans. So they're their own branch on the family tree. Not a different domain, but we're not sure if they're a different kingdom or a different phylum just yet. Euryarchaeota, what domain? Archaea again. Why is this group in that domain? It's the rRNA sequence again. The rRNA sequence is what put any organism into any of the 3 domains that exists. These guys, both of these guys are prokaryotic. They don't use peptidoglycan to make their cell wall. They're not bacteria. They're archaeans. They're their own little separate group. Weird little side note on the Euryarchaeotas, a lot of them are halophiles, which again means they like when things are really, really salty. That completes chapter 10. I wish I could give you more specifics, but like I said, everything within classification is sort of in flux and has been for a while. So just try to keep up with current events and current research papers if you're really interested in the taxonomy of different organisms.